Effector T-cell interactions with target cells are initiated by antigen-nonspecific cell adhesion molecules. Once an effector T-cell has completed its differentiation in the lymphoid tissue, it must find target cells that are displaying the MHC peptide complex that it recognizes. Some Th2 cells encounter their B-cell targets without leaving the lymphoid tissue. However, most of the armed effector T cells emigrate from their site of activation in lymphoid tissues and enter the blood via the thoracic duct. Because of the cell surface changes that have occurred during differentiation, they can now migrate into tissues, particularly at sites of infection. They are guided to these sites by changes in the adhesion molecules expressed on the endothelium of the local blood vessels as a result of infection, and by local chemotactic factors. The initial binding of an effector T cell to its target, like that of a naive T cell to an antigen-presenting cell, is an antigen-nonspecific interaction mediated by LFA1 and CD2. The level of LFA1 and of CD2 is twofold to fourfold higher on armed effector T cells than on naive T cells, and so armed effector T cells can bind efficiently to target cells that have lower levels of ICAMs and LFA3 on their surface than do the professional antigen-presenting cells. This interaction is normally transient unless recognition of antigen on the target cell through the T cell receptor triggers an increase in the affinity of the T cell's LFA1 for its ligands on the target cell. The T cell binds more tightly to its target and remains bound for long enough to release its effector molecules. Armed CD4 effector T cells, which activate macrophages or induce B cells to secrete antibody, must maintain contact with their targets for relatively long periods. Cytotoxic T cells, by contrast, can be observed under the microscope attaching to and dissociating from successive targets relatively rapidly as they kill them. Killing of the target or some local changes in the T cell then allows the effector T cell to detach and address new targets. How armed CD4 effector T cells disengage from their antigen-negative targets is not known. Although current evidence suggests that CD4 binding directly to MHC class II molecules on target cells that are not displaying specific antigen signals the cell to detach. And that's effector T cell interactions with target cells are initiated by antigen-nonspecific cell adhesion molecules. Have a great day.